Voy a salir en YouTube. No, no, no. It's okay. Hi, everyone. Let me introduce myself once again. I'm Mauricio Garcia Godinez, and today I'll be working and talking to you about my project, which is buttermilk characterization for product development. So first of all, what is buttermilk, right? Buttermilk is a byproduct of butter productions, which is a source of phospholipids, proteins, minerals, and vitamins. Yearly in Mexico, around 43,000 tons of butter are produced. And all the buttermilk that is produced with this butter is discarded. Now, what does it imply? This implies, sorry, someone in Zoom saying something? No? Okay. So what does it imply? As I was saying, this implies water pollution. Now, it's a lot of production, a lot of mass production, and this implies an impact in the environment. Okay, so buttermilk is normally used on the confectionery industry. For the ones that doesn't know what this is, it's basically making the cakes, desserts, cupcakes, all that. And in some countries, they ferment this product and make beverage of it. But why buttermilk? Regarding that it will have benefits from the environment, since it's a waste, it also have benefits due to the phospholipids that it has bioactivity that have the potential to be anti-carcinogenic, especially to colon cancer, protective against bacteria, toxins, and infections, obesity, diabetes, and cardiovascular disorders. Now, this potential has to be like with more detail into more research, but this has the potential to be health, to increase the health of the consumers. Now, what is the objective of this project? First of all, to characterize buttermilk obtained from commercial acidified cream, and to use a new product formulation with buttermilk as main ingredient, with the potential with environmental and economical impacts. Thank you. Now, how do we extract the buttermilk? For this, we follow the traditional method of wiping cream. Now, this process takes around 15 minutes. Now, as we can see in this image, we can see a phase separation between a solid and a liquid one. We will continue the whipping until there's a clear phase separation. Now, what we do with the butter, that is the solid phase, we're going to use a cheesecloth, and by pressing with the hand, we're going to separate the butter from the buttermilk. Now, all the butter that was retrieved from this experiment was used for further other experiments in the chemistry lab. Now, for the characterization of it. For the protein content, we use Bradford. For main protein fractions, we use SDS-page, reducing sugar DNS. We basically dehydrate the sample. Lipid content, we use lactoscan. And lastly, we use density. For the results, as we can see, the protein in the pellet of the buttermilk and in the supernatant is basically the same, which is interesting because we were expecting to have a bigger difference among them. And it actually kind of be really close to the reference values, which I will talk in a little bit later about those. For the reducing sugars, now we can see a bigger difference. For the reducing sugars in the supernatant, we can see that not even half of the reducing sugars in the whole buttermilk, which is interesting because when we are planning to make a product, we can choose if we can use the whole buttermilk of the supernatant depending on the amount of sugars that we want. In this case, we use the whole buttermilk. There is no reference value because we couldn't find any reference that use the same methodology or with the same principles that we use to have a clear comparison. For the solids percentage, as you can see, is 12.14. For the lipid content is 2.2. Now, what happened with the reference values here? We have to remember that when we talk in dairy products, cows create a lot of uncertainty. It depends on the variety of the cow, the way the cow was treated, the food that the cow was supplied with, and social factors such as the age of the cow and others. So this may be an explanation of the difference between the values that we obtain throughout characterization and the values that we can find on literature. For the one of the SDA's page. Okay, so the ones that we are interested in is on wells number 6, 8, and 9, which are the samples that we use. Now, I don't know if the people in Zoom can see it, but here's a band on well number 6 that is the supernatant that will correspond to protein lipids that are capsaicins. Now, also, and this maybe is not seen in this picture, but we have other 2 bands that are very really here. The one that should be here corresponds to beta lactoglobulin, sorry, and alpha lactalbumin, which are proteins that are mainly present in dairy products, which is a good sign. It indicates that we're doing a good job through the characterization. Now, on the bottom example, we can see more likely the capsaicins, but we may not see any of the other alpha and beta proteins. And on the 9, that it will be the pellet of the buttermilk, we actually can see there is right here the beta and the alpha. Thank you. Now, which product we developed? We developed a dressing salad. Why a dressing salad? Due to the characteristics of the buttermilk and how it's more manageable and how it's more easy to work with. So as you can see, the main ingredients are buttermilk, as it was proposed, green lemon juice, pepper, garlic sand, just to add flavor, santan, and Arabic gum, which for the conservation of the product, and different aromatic herbs and cream cheese just to enhance the flavor. Now, once the product was through pasteurization, we still need to check that it's safe for consumption. We follow the Mexican law, that is this one right here, created in 1979, that is used for the detection of microorganisms that may be pathogenic for humans. As we can see, here's one of the many replicas that we have from the Petri dishes. This one are for fungi. This is PDA. And as we can see, there's basically nothing, which is a good sign. Here we go with the yeast, which is the same, nothing, which once again, it's a good sign. And would you please? Thank you. For the fecal coliforms and E. coli, we can see here some little bells. And in these bells, we cannot see any bubbles, which is a good sign. If we have bubbles, that will indicate the presence of either a fecal coliform or E. coli, which will indicate further specification. And lastly, we have for salmonella, which is also really bad. But once again, we have clear and empty Petri dishes. And sadly, we didn't have enough time to go through sensorial evaluations, but for the ones that are brave enough, and as you can see, the product have nothing growing on them, we have right here the sample of the product for the ones who want to taste them. And lastly, I want to say thank you to some classmates that helped me through this entire year of the production of this product, which is Edna Arciga, Miguel Adan, and Juan Salvador. And of course, to my mentor, Dr. Manuel Santos Basurto, who helped me through this entire year. Thank you.
Thank you, Mauricio. Do we have any questions from the audience or in Zoom?
Yes.
Are you planning to go on with this one with the production and then trying to open it for the market you really have the plan and how it is working that. And now, which one is the timeline, more or less?
Well, I think it would be a shame not to continue with the product, even though that we already spent almost a year designing it. And I've been studying, like, since it's a product, we just doesn't need like the STEM part. We also need like the business part. So I did like a really brief and like have a notion of the market. And according to INEGI, around 3% of all Mexican salary is destinated to salad dressings. So I think that is a good market and it has a really good opportunity. And since the flavor is really similar to other salad dressings, but with like small differences, I think it will be a good product for the market since it's not going to be like that shocking for other people. And I think it's going to be good accepted. Well, for the long term, I still have to make the sensorial evaluations. I still need to do more market research. Honestly, I'll say at least other semester, but focus more on the how to bring it out to people more than just in the lab.
Mine is like a really kind of simple question. I was just wondering like how long does it last? Because I think you didn't add any like preservatives or anything. So like about how much like on the fridge, how much is it still good to go, you know?
Thank you, Jimmy, for your question. And we add some conservatives, is the potassium sorbate, as the one to avoid just the decomposition of the product. And from now on, with the sorbate and with the other, obviously, other that we have used in the past, roughly 3 months once it was opened. And either all those products didn't have the pasteurization process. So maybe with the pasteurization process and everything, maybe even longer, maybe 5 months, 4 months. Sorry. And any further questions for the guys from Zoom? Ah, okay. Is that one right? Okay. What was the final product achieved by saving water? Ah, sorry, I misunderstood the question. What was the final product achieved by saving water? I don't know if I'm getting correct the question. If the product is going to save water in the future, I don't know if that's the question. Yeah, the thing is when we are producing butter, normally they don't do the traditional wiping cream that we use in the lab. They use other acids and other chemicals in order to even like have a higher yield on the butter. So all that contaminants are with the buttermilk and the water. So by doing this, we basically have no waste on the water pollution. So yeah, that was like the goal and kind of achieved it. Any further questions? Thank you, everyone.
Thank you, Mauricio. To continue with our presentations, we have undergraduate senior student Luis Humberto Villasana, who is going to talk to us about evolutionary study of cyclin-dependent kinase 1.
How long can I speak?
10 minutes.
How long?
10 minutes.
Oh, long.
No. Oh, no. Everybody in Zoom is going to be listening to you without.
Can I start?
Okay, hello everyone. My name is Luis Humberto Villasana Avila, and I'm going to present about the evolutionary study of cyclin-dependent kinase 1. Could you change slide, please? Okay, first I divided the presentation in these sections: introduction, objective, methodology, results, conclusion for the work, acknowledgement, and references. So the introduction, I had for this project to select one protein of my choosing that I would like to study is key amino acid residues. the protein that I choose was cyclin-dependent kinase 1. Why did I choose this protein? Because I was interested in its properties. As you can read, CDK1 has the property that it is key for the function of mitosis and meiosis, for the transition and its regulation. So its overexpression can lead to cell proliferation that can lead to cancer. That's why I'm interested, because I want to be a genetic counselor and I'm interested in this kind of phenomena. And its inhibition can damage normal population cells, which can damage the cells. Okay. So the objective, as I mentioned, of the project was to identify key amino acid residues involved with the function using evolutionary tools that I'm going to explain in the next slide. So I'm going to mention the methodology step by step, how did I did it, and I'm going to briefly explain it. Okay, first I grab the sequence from NCBA, which is CDK1, the sequence of the protein. Then I use Hammer. Hammer, I use it instead of BLAST, and Hammer, it is a program that lets you find other sequences similar to the one of your choosing, but instead of choosing the sequences that are similar by amino acid sequences, its criteria is by homologous properties. So it focuses more if the protein has the same properties of your protein of your choosing that the sequence itself. Of course, it also comes by the sequence, but it is more focused on having the same homologous properties. Then there is math. From Hammer, I got 100 sequences in total, and then I use math for multiple sequence alignment because I wanted to because I needed to align them in order to study them. Then I use Mega 11. Mega 11 is a program that you can download in order to identify which phylogenetic tree model I was going to use in order to create the tree, because I was going to use the methodology of a maximum likelihood. And to use that methodology of phylogenetic tree, you need to know which model you're going to use, and you are not able to just randomly choose which model you're going to use. You need to use Mega to identify the model. Then I use PhyML because with Mega I already identified which model I was going to use in order to create the phylogenetic tree. Well, PhyML lets me create the phylogenetic tree already because I already identified which model I was going to use, which was L plus G. Then I use ITOL. ITOL is a program that lets you change per se or modify the phylogenetic tree to make it more appeal to the eye and make it more visual. So it is easier to identify different properties or characteristics that I'm going to showcase later. Then I use FireProt ASR. ASR is from Ancestral Structure Reconstruction. What does this mean? I use FireProt ASR with back engineering to recreate the amino acid sequences from the phylogenetic tree. I managed to recreate the amino acid sequences of all the ancestors of the phylogenetic tree. I'm going to also showcase this later. iTaser, when I had the amino acid sequences of all the ancestors of the phylogenetic tree, I choose 5 ancestors that I wanted to study and use iTaser, which is a server that lets me create like a simulation of the 3-dimensional structure of the protein. Then I use Mystic. What is Mystic? Mystic is a server also, or a program that lets you using the sequences and a model as a reference to understand the coevolutionary amino acids of a multiple sequence alignment. I'm going to also explain this later. And finally, PyMol, because PyMol is a program that lets me visualize better the three-dimensional structure, so you can observe better which are the key amino acids, that I identified with MYSTIC. And I also going to show you this later. Okay, the results. Here is the phylogenetic tree that I managed to modify with ITOL. How did I modify it? I only added color and I make it so that the tree has the color. So you will visualize better the characteristics. What characteristics? I modify the colors so you can identify the 100 sequences depending of the kingdom. Red is the kingdom of fungi, blue is the kingdom of animal, green is the kingdom of plant, and purple is the kingdom of protists. Now, what does this mean? This phylogenetic means, well, first of all, here at the center is the common ancestor of all of these sequences. Then it is read from left to right. And for example, here you can ascend. Let's say you want to read this. This sequence is before than this sequence. What does this mean? That this sequence, for example, this sequence is the evolutionary of this. It's a more evolutionary. Has evolved more than these sequences from here. And the most involved protein from all the 100 sequences is ROT1. Okay, so I mentioned to you that I use iTizer to reconstruct three-dimensionally structures because I wanted to showcase their similarities and how they have been evolved with time. Here, you can see by color which ancestor corresponds to which section of the phylogenetic tree. The dark blue is the one over the left, and the orange is the point that is colored in orange, and so forth. I just want to look at it a little bit and try to distinguish the similarities and difference between these proteins, because they shall have similitudes, but also some difference because it has been evolved with time. And I also wanted to let you know that this protein, as I said before, is crucial for mitosis and meiosis and its regulation and cycle. So this protein is in almost all of the kingdoms and it's essential for life. Here in red is the key amino acid residues that I identified using this coevolutionary map from the server MYSTIC. And basically, the ones that are in red are, I know that coevolutionary because they are the more conserved. And I use the multiple sequence alignment to create this histogram to identify which one are they repeat the most and the ones that have evolved with each other. And in conclusion, it is important to be able to use evolutionary tools to understand the key amino. I was able to use them to understand the key amino acid residues, and this will be important to further the work and be able to understand why a protein has its function and who is able to perform their functions. And if we want to study further, we can do it. We can do it in the laboratory to confirm the results of my project. And I just want to acknowledge Dr. Bernardo Perez Zamorano, who is here, and the great CQ-AState coordination staff and the Kansas State University campus Querétaro.
Do we have any questions from the audience or the audience on Zoom?
There's Dr. Medina raising his
Dr. Medina, feel free to ask your
Thank
question.
you. Can any, well, first, Luis, extraordinary work. It's a lot of tools that we need to use for one protein. Can any of these tools can give us the quantitative measure of the protein?
The quantitative measure, can you explain a little bit more so I can understand the question?
By numbers, not if there is the protein or not. So it's if you have any numbers to know how much protein do you have, how many
Well,
proteins.
I don't know if I understand the question, but I have 100 sequences. So I have 100 proteins sequences which I work with. And I don't know if this answers your question, but it will be better to use more sequences to have a better understanding of the evolutionary traits of the amino acid residues. I'm not sure if this answers your question.
If we have a solution with of a patient or a blood test, can we know how many proteins do you have in there?
No.
No. Okay. Thank you.
So just thinking out loud or wondering, do you know if any of these amino acid that co-evolved within this tree that you built are the ones responsible for the restriction in terms of the cyclins that CDK1 is able to bind? Or are they in other regions like the regulatory phosphate binding region or ATP pockets or such and such? Or do they have any functional impact in terms of the structure of the CDK1? Or is that something you can't tell at this stage?
Okay, thank you for the question. I would like to say that sadly, I cannot answer that question because, as I said, I was only able to identify the key amino acid residues. So one of the further works that we can do is to do that, to identify which sections of the protein are the ones that bind to other structures or other proteins to do the cascade of the regulation cycles. But it can be done with binding simulations in dry lab, or it can be done studying in the wet lab.
Not a question.
How could you use the CDK1 protein in human therapy? Thank you for the question. CDK1, as I said, it can be responsible for cell proliferation that can lead to cancer. So we can use CDK1 if we understand its properties, which key amino acid residues are responsible. We may be able to identify which mutations are damaging for the cell cycle, and if we are able to identify these mutations that lead to cell proliferation and damage the production of the cell cycle, we may be able to use genetic therapies to modify the proteins and be able to correct this. Maybe in the future, I don't think we can do this right now, but we may be able to in the future to modify the protein and be able to maybe not cure a patient from cancer, but maybe to slow down the process. And if there is a genetic therapy from a fetus and the fetus shows that their CDK1 protein structures, the CDK1 protein sequence it has mutated to be damaging, maybe we can change the sequence to make it that they produce healthy proteins that regulate correctly the cell cycle.
Well, thank you, Luis, for this amazing presentation. Can we give him a round of applause, please? Next is undergraduate senior student Jacqueline Romani Vargas, and she will tell us about production of specialized metabolites with potential health applications in hairy root cultures of Anato.
Hello, can you hear me?
Yes, we can hear you.
Perfect. Can you tell me if you can see my screen share?
Yes, perfect.
Excellent. So, hello everyone. I'm Jacqueline Vargas. Some of you might know me, but for those of you who don't, I have been on exchange at A-State Jonesboro for the past 2 semesters. And during this time, I have been working at the lab of Dr. Fabricio Medina Bolivar at the Arkansas Biosciences Institute. And this is my research project on the production of specialized metabolites in hairy root cultures of the plant annatto. So there are several diseases that affect humanity for which we don't have effective enough treatments yet. And one approach to solving this problem is drug discovery. One avenue for drug discovery are specialized metabolites. These are molecules produced by plants under certain conditions, such as stress. We specifically focused on the plant Bixa orellana or annatto. We also call it achiote in Mexico. This is a plant native to South America that is commonly used as a spice and food coloring. It is used in many Mexican traditional dishes, and it is responsible for the color of cheddar cheese. In traditional Latin American medicine, it has been used for its medicinal properties to treat burns, infections, inflammation, high blood pressure, high cholesterol, and prostate disorders. It has also shown anti-malarial activity. So we were interested in this plant because of its medicinal properties, and we think that they might be due to the terpenoids that it produces. Terpenoids are specialized metabolites produced by some plants in response to stress. So just like we produce cortisol when we are stressed, this plant produces terpenoids when it becomes stressed. But we didn't use the whole plant. We used only the roots, and specifically a type of plant tissue culture called hairy roots. Hairy roots are immortalized roots. By this, I mean that they have indefinite and rapid growth. A great advantage of hairy roots is that they can be grown in liquid media, as opposed to growing them in the soil or other solid medias. They also release the compounds into the media, which makes purification way easier. And they are not affected by environmental factors such as weather or water conditions. So you can grow these hairy roots all year round inside laboratory conditions. And they grow inside these cute little flasks. So now let me talk to you about the basics of hairy root culture and how we establish a hairy root line. So for these, we take the leaf of the plant and infect it with a bacteria called Agrobacterium rhizogenes. After the infection, roots start growing on the wound. And after we have enough roots, we can detach them from the leaf and transfer them to a flask. Once we have enough flasks with roots, we can start stressing the roots with chemical elicitors. When they become stressed, they start releasing the specialized metabolites into the media, and then the media is purified to obtain these compounds of interest. So the objectives of my project were to induce the production of terpenoids in the hairy roots of annatto or achiote, and to establish a sustainable terpenoid production platform. This was the methodology that we used, and we will first focus on the first half. So we start with subculturing, or the process we do to obtain enough flasks of hairy roots. Once we have enough roots, the old media in which they were growing is removed, and we add new media containing the elicitors or chemical stressors. After 8 days of stressing the roots, we can collect the media and purify it. But before I show you the purification process, I want to show you the subculturing, which was very interesting to me as it was something that I had never done before. So this is how we obtain many roots. You can see me cutting the tips of roots around 10 to 12 and transferring them to a new flask. So from every flask, you can obtain around 4 to 6 new flasks, and that's how we obtain a lot of new plant material. And this is the growth time lapse after the video that you just saw. This is 3 days later, 6 days, 10 days, and 14 days later, time at which they are ready to be subcultured again. Okay, so now that we have enough roots, we can start the elicitation process. In this process, 12-day-old roots are stressed with the elicitors, time at which they stop growing and start producing defense compounds, the terpenoids. The elicitors or stressors that we used are hydrogen peroxide, which induces oxidative stress, magnesium chloride, which is a cofactor for the enzymes involved in the biosynthesis of these specialized metabolites, methyl jasmonate, which is a transcription inducer of the genes involved in the biosynthesis of the specialized metabolites also, and methyl beta cyclodextrin, a compound that prevents feedback inhibition by encapsulating the compounds as soon as they are produced. So as I mentioned before, after 8 days of stressing the roots, we can collect the media in which they were growing for purification. And here I want you to see the control versus the elicited roots. So in each of the pictures, you can see to the left, normal, happy roots, non-stressed, and to the right, stressed or elicited hairy roots. And we can see from the color of the liquid that there is clearly something being produced and released into the media. 8 days of stress, and we collect the media for its purification. And then here is the extraction and purification workflow. We take the media and do solvent extraction using ethyl acetate. We separate and obtain the organic phase, which is then analyzed through HPLC. Here are the results of the HPLC. And as you can see at the top, we have the control, the happy non-stressed roots that did not produce any compound. Nothing was produced for them. However, for the elicited or stressed roots, we can see the chromatogram at the bottom showing 6 different compounds that were produced. Here are the 6 compounds with their respective absorbances. So we can conclude that after elicitation, 6 compounds with similar UV absorption spectra were identified. And the work that I performed with other lab members at this lab will allow the identification of novel specialized metabolites with potential health applications from achiote or annatto hairy roots. So regarding future research, I will continue with the project thanks to the fellowship to a summer research fellowship by the American Society of Pharmacognosy. This will provide me with funding to continue the work that will involve purifying the compounds produced by the achiote or annatto hairy roots and characterizing them through NMR and mass spectrometry. So I just want to say thanks to Dr. Fabricio Medina Bolivar, Dr. Amit Rag Sharma, who was in charge of teaching me a lot of the techniques involved in this project, and to my direct supervisor, the graduate student Gaurav Gajral, and to the rest of the lab members of the Fabricio Medina Bolivar Lab, to A-State Querétaro, A-State Jonesboro, and the Arkansas Biosciences Institute. So now I'm happy to take any questions that you have.
Thank you, Jacqueline. Are there any questions?
Hello, Jacqueline. I'm asking if it could be possible that the roots that were stressed could be propagated with the method you did so that they produce the terpenoids without any of the needed stress?
Oh, that's a great question, actually. So the roots only start producing these compounds when they are stressed. So once they stop being stressed, they don't produce anything anymore. So it's like, as I said before, when we are stressed, we produce cortisol. When we stop being stressed, we stop producing cortisol. So it's not a process that once you stress them, they will continue producing specialized metabolites forever. No, it doesn't work like that. So we need to stress them each time.
There is another question on the Zoom chat. It says, are you considering that your approach has the bias that you are only considering the roots to extract the bioactive compounds?
So I'm guessing the question is regarding, since we are just focusing on the compounds produced by the roots, we are ignoring all of the other possible compounds that can be produced by the plant. That's what I'm assuming. So in this case, we have, although this Bixa Orellana project is relatively new with these elicitors, a lot of research has been done on hairy roots of another plant called peanut. And from these, we have noticed that by stressing the roots with certain elicitors, they produce the compounds that we are interested in. So in our case, in our project, we are interested in only isolating terpenoids. Although it would be good to study all of the possible compounds produced by the plant, we are right now just focused on terpenoids, and we know our protocol works for inducing the production of terpenoids.
Yep, there is another question here.
Thank you. Hi, Jacqueline, Dr. Belmont here.
Hello.
I have a question about the extraction process in order for you to improve if you want to make this like a viable project in order for production of the terpenoids. I believe that the extraction is a crucial part. Speaking about the cyclodextrins that you're using, do you know which is the size of the cyclodextrin that you're using? And have you think about modifying that cyclodextrin in order to improve the extraction?
Well, so after the roots are stressed with this combination of elicitors that includes the cyclodextrin, we purify it through first column chromatography, then reverse column chromatography, then HPLC to isolate each individual compound. Right now, what I presented was just like a general extract showing all of the compounds that are produced. But when we are actually into working in the purification, we do several steps that separate very specifically each compound that we have produced. And regarding the size of the methyl beta cyclodextrin, I don't know the exact molecular weight. I just know that it's a very big molecule that has methyl groups inside that trap non-polar compounds.
Before closing up, hello, Jackie, this is Dr. Katy.
Hello,
From
Doctor.
our side of Campus Querétaro, we would like to congratulate you for winning the fellowship, and we hope you're gonna enjoy this summer over there also.
Thank you very much, for sure. I'll do my best to represent Campus Querétaro in a good way.
Thank you.
Thank you.
Okay, now we will proceed with a presentation under the title Interactions between BCL2 proteins and novel curcuminoids, a molecular docking and DFT analysis. The undergraduate senior student Edna Lucero Arciga Contreras presents the analysis of the interaction between the anti-apoptotic BCL2 protein and curcumin. Her mentor was Dr. Fernando Belmont. Thank you.
Hi, everyone. I'm going to present, as you already said, the interaction between BCL2 proteins and curcumin, and we did a molecular docking and DFT analysis. This project was for El Verano de la Ciencia Región Centro 2022. These are the table of contents that we will be going through the presentation. So for starting, BCL2 proteins are proteins that regulate cell death by mitochondrial outer membrane permeabilization. These are anti-apoptotic BCL2 proteins that are highly expressed in cancer cells. There are studies that show that these are related to the development of chemotherapy resistance. On the other hand, we have curcumin, that is a molecule extracted from Curcuma longa L, and it has important antioxidant properties. It is a highly symmetrical hydrophobic molecule, and it's being studied as a potential anti-cancer agent. So why is this important? So in cancer cells, we have the overexpression of anti-apoptotic BCL2 proteins, which means it doesn't let the cancer cell to undergo cell death. And in this, we have cell proliferation and cancer development. However, there are studies that show that when cancer cells are exposed to curcumin, there's an inhibition of the anti-apoptotic BCL2 proteins, and there's a promotion of pro-apoptotic proteins that inhibits these proteins and then will inhibit the proliferation of cancer cells because it leads to cell death. From the methods we used, we have molecular docking. That is a computational method that analyzes the behavior of a molecules on a protein's binding site. So we have our target molecule, that in this case is the protein, with a ligand, that is the curcumin. And by docking them, we generate a complex where they are interacting with each other. Then we have the density functional theory, DFT, that is based on quantum mechanics, that uses Oppenheimer's approximation to solve the Schrödinger equation. And the notch elastic band method, it's really good for calculating transition states, and it finds the minimum energy path between reactants and products. So the objective of the project is to investigate interaction between the 6GL8 BCL2 protein and the inhibitory action of curcumin through in silico models, combining bioinformatics and DFT. So the first thing we did was calculating the equilibrium constant of curcumin because it has an ketoenol equilibrium, and we wanted to know which tautomer is more present in solution. After that, we did the optimization of curcumin using Orca software, and we used the functional BP86, the F2SV basis set, the B3BG empirical dispersion method, and the CPCM solvation model of water to imitate biological conditions. And we also did the optimization of the BCL2 protein with the Yasara server. After we had the optimization, we generated the complex with Autodock tools, and we used site-specific docking. That means that we didn't let the curcumin interact with all the protein, but just in the active site of the protein. When we had the complex, we extracted the residues showing the formation of hydrogen bonds with curcumin to study them as a separate system. And lastly, we had the calculation of the transition state using an FTS. We also used Orca, and the same conditions as the optimization were established. So as for the results, we had the curcumin interacted in the active site, and we observed that it formed hydrogen bonds with glycine, arginine, and aspartate, all of them from chain A of the protein. And we got an equilibrium constant of the enol form being 1,000 times more present in solution than the keto form. So we just studied the interactions of the protein with curcumin in the enolic form. We had an inhibition constant of 394.33 micromolar and a binding energy of minus 4.64 kilocalories per mole. When we observed the complex, we proposed a proton exchange in which an oxygen from aspartate deprotonate the phenol of the curcumin, and with that proton exchange, the system was left with a negative oxygen and a positive nitrogen from arginine. However, when we calculated the transition state, the proton exchange didn't happen because the system was not basic enough to accept the proton. So what we observed instead was that glycine came closer to curcumin, and it generated another hydrogen bond with the enol. So it moves, indicating a conformational change in the amino acids, and it surrounds a little bit curcumin, and also the curcumin bends a little bit more. As we can observe, the yellow molecule is the reactants, and the blue are the products. So as we propose a proton exchange that didn't happen, we increase the interactions between the molecules. And for the energy diagram, we observed a thermodynamically favorable interaction, as we obtain a free Gibbs energy value of minus 4.67 kilocalories per mole. So we conclude that the tautomer preferred in the keto-enol equilibrium is the enol tautomer, and that we observe a thermodynamically favorable interaction from the proposed proton exchange that resulted in a conformational change of the protein that possibly suggests the inhibition of the anti-apoptotic BCL2 protein. This work was supported by the technical staff and the Laboratorio Nacional de Visualización Científica Avanzada from UNAM. And I would like to extend a special acknowledgement to Dr. Bellman for his guidance and mentorship through El Verano de la Ciencia.
Thank you, Edna. So does anybody in the audience has any question for Edna? Yes, Victor.
Okay, so my question is regarding the approximation that you are using to solve the Schrödinger equation. I don't know if you could explain what is this approximation. So what does it mean? And if you know, for instance, what are the conditions that you need to meet to actually use the approximation, and also if the molecule that you are using meets precisely those restrictions in order to use this approximation that you're using.
So we are using the Oppenheimer's approximation because it's related to quantum mechanics, and we wanted to calculate interactions between atoms in the system. And as this approximation gives us numerical results that we can interpret and use, that's why we use it. And before starting with the calculations, all of that conditions were met, as well as the Lipinski's rules and all of that.
Thank you. Any other question? Yes, Dr.
I'm asking, how will you continue this work? I guess that you had pretty good results from the quantum mechanics part. Are you planning to do some experiment in the lab to confirm that really you have these anti-apoptotic results?
Before doing the actual lab work, we wanted to change the solvatation model with for having a lower isoelectric point, but we wanted first to confirm that the proton exchange and the interaction actually happens in water because it's a hydrophobic molecule, but after that it will be possible.
Thank you. Any other question here or online on Zoom? I think we have one here. Is the strength of a hydrogen bond sufficient to modify the protein ligand interaction and inhibit the BCL2 protein?
I think so, because hydrogen bonds can create good networks, and also we are observing a conformational change. And in terms of protein function, the conformational state of the protein is very important for the function. So if we're observing differences in the conformation of the protein binding interactions, it can suggest that it will inhibit the protein because, and also the curcumin is in the active site, so it won't allow anything else to bind to the protein.
Thank you. Another question here or in Zoom? Okay.
Yeah, we have time.
Yeah, we actually.
Oh my. Thank you. Okay, and, it's been a very extensive work, as I can see. So I just want to continue a little bit with Dr. Velasquez's question that has to do, of course, I'm a physicist, so I find that very interesting. You do talk about using this technique of density functionals theory and so on, but I don't think it was particularly clear what assumptions are made. So you did mention that you use the Oppenheimer approximation, right? Precisely what does that approximation say? What is the key assumption that you make? I'll walk because it feels more like it should be a dialogue. So I promise I'm leading you somewhere. So precisely what are you assuming?
When we are using the Oppenheimer's approximation, we have a wave function in terms of electron positions in the systems and nuclear positions, and that's where we are.
Exactly. So you are assuming, so you're assuming that you can actually decouple, no? The wave from the nucleus versus the wave from the electrons, no? These type of things you need to make, I think, particularly clear when they ask you what are the assumptions that you are making. I think that was the direction of, or at least one of the directions of the question. Is that correct? Yes. And also, what are the limits of the theory that you're using? Because, as you said, you're making certain assumptions, right? Is this functional theory appropriate, for example, to talk about transitions in which things are thermodynamically preferable and so on? So I don't doubt that you understand these things, but it's important to tell them to people, okay? Because you've done a very big work, so you might as well showcase it, right?
We have a raised hand on Zoom, so please, Dr. Juan.
Hi, thank you. And did you measure the time of this change in the molecule? Or it's forever, this change?
Well, I don't know if it's forever, but we had a favorable interaction, so it's not that probable that it will detach that easily. But from the results we have, we don't know if it will be binded forever and make the protein like that doesn't function anymore. But if we do have the inhibition, the cell will go to cell death. So then it will be like not forever, but the cell will be dead. So
Okay, thank you very much.
Excellent, thank you. Now we will proceed with a topic of theoretical analysis on the reactivity by bis and PA esterification over resveratrol application of DFT for reactivity indexes. Undergraduate senior student Miren Ugarte Maldonado presents the consequences of selected esterification over resveratrol in the reactivity indexes of the HOMO LUMO gap, chemical potential, chemical hardness, and Felicity, among others, by calculating the energy available on the FOM by applicating TDDFT calculations. Her mentor was Dr. Fernando Belmont.
Okay, well, hello everybody. My name is again Miren Maitane Ugarte Maldonado, and I'm going to present the theoretical analysis on the reactivity of BCMPA esterification of a resveratrol with this application for DFT and reactivity indexes. So what is resveratrol? Resveratrol is a naturally occurring polyphenol compound, that is this one. It is found primarily on plants and forest fruits. It's being generally used for, it's been produced for defense against fungi, stress, and especially oxidative damage in these plants. It was first discovered in 1975 in grapevines by Drs. Lancake and Price. And well, there are two main conformations of this molecule. It can be the cis or the trans conformations, but through thorough analysis, we have found that the most common available nature is the trans one. So we will be focusing our investigation in this one. Also, it has been traditionally used in Oriental medicine, especially in Japan by the Kojo-un roots, which is used for its beneficial applications. They are thought to be anti-tumor, antiviral, and especially antioxidant. And well, we diving into its natural reactivity and that of induced with esterification. But although it has seemingly great biological applications, its biodisponibility is very low because it doesn't solubilize in water. So, yes. So we're gonna take from the perspective of frontier orbital theory. This is a very valuable model for assessing reactivity in an initial approach, like the first step in this matter. Because rather than focusing on the total electronic density, we are just focusing on the HOMO and the LUMO surfaces. They can be used in understanding pericyclic reactions, such as the Diels-Alder that we know, where the electrons found on the HOMO region flow through charge transfer to the LUMO region and bonds are formed. Well, this theory was further amplified and deepened by Fukui in the 1950s, which he used for the description of the reactivity region inside the molecule. So DFT again, we use this quantum mechanic tool to define the energetic interactions of both the resveratrol system and the new molecules that we proposed. This is a computational methodology because doing this math manually would be very difficult. It focuses on the transition from the many body problem perspective that it basically and transform it into a DFT perspective of a whole density and a similarly simple area to work with. It has a very high computational power demand, and it's like proposed for professional use. And well, it also helps us with potential candidates for our study to understand them through predetermined chemical behavior. So what is our objective? Our objective was to evaluate the reactive properties of resveratrol and BCMPA, derivatives through the assessment of the reactivity indexes and the plotting of electronic and orbital surfaces. So for our methodology, we work with these initial candidates in the software Gaussian 16. We use a B3 level of theory, DF2 basis set, and a G3BJ empirical dispersion, which basically enhances our results because it compares against pre-existing databases. And well, it's important to highlight that all these calculations were done with solvent as with water as a solvent, because its final application is towards biology. Okay, so what are our activity indexes? They are meant to assess the activity of a molecule within a certain framework, and they have both differential and numerical solutions. These are the numerical solutions we use because we work with the HOMO and the LUMO surfaces. We have the HOMO-LUMO gap, which is basically the energy difference between both surfaces. We have the chemical potential, which is the feasibility to exchange electronic density. We have chemical hardness, which can be thought of somewhat opposite because it's the resistance to alter this electronic density. Electrophilicity index, the stabilization energy when a system gains an electron. The change in Nmax, that is the maximum numbers
an electrophile may gain. And well, the N relative, that it's a measure of nucleophilicity compared against resveratrol, that is our main focus. So for our initial candidates, we wanted to test these reactive properties in different molecules. So based on previous work done by Dr. Belmont, we wanted to modify this core molecule by esterification. We esterified 1, 2, or either 3 phenols in this molecule with this MPA. They could be in a protected version. Here is the esterification, or in the unprotected manner, that is this here. Okay, so in order to unbiasedly evaluate all these molecules, because well, you see there were a lot, we use the reactivity indexes I just mentioned. So for the HOMO-LUMO gap, we wanted the lowest value that it corresponded to B's, the B's protected. Only 1 phenol was protected. For the chemical potential, we got rest as the winning molecule with a less negative value. We wanted a lower hardness, that again is B's per rest, and the highest electrophilicity in max and in relative, which won respectively B's, res B's, and resveratrol. So we now had 3 competing molecules, each hitting 2 scores, but we wanted to elucidate the results a little bit more. So we added an extra marker or index, that it was solvatation. And we can see that the B's, those res B's, was significantly more soluble in water than its competitor against resveratrol. So we then took these results and wanted to understand a little bit more of how these molecules work. So we added the indexes of electron accepting power, electronating, redox absolute, and in respect with resveratrol. The B's, those res B's molecule had higher electron accepting power, but it also had a more negative redox. That might sound contradictory, but we found that this value corresponds to the difference between the redoxes of hydrogen and iron, which was a great discovery. Also, we, when thinking deeper about these results, we associate them with the chemical hardness that we saw previously. So with all these values, we could only assume, well, catalog these as a radical scavenger rather than an antioxidant due to the combination of all the indexes they marked. And well, it is thought that they would neutralize the radicals by creating covalent bonds instead of just releasing the electron to the existing radical as its counterpart resveratrol would do. So we then wanted to see where this activity was located, and we plotted these potential maps. That's one resveratrol, and the B's, those res B's molecules. As you can see, the electronic density are almost equally distributed, and we cannot elucidate a lot of this from the images. So we dug deeper, and we used the Fukui functions. Here we proposed an electrophilic attack for the resveratrol and a radical attack for B's, those res B's, but it's important to highlight that in order for the electrophilic attack to happen, this condition needs to be supported by the fact that if a radical behavior matches that of an antioxidant agent, then it will be thought as an electrophilic attack. So here we can see that the main place for the electrophilic attack to occur is this phenol, and it's located. It's highly concentrated and located. We see and also a little bit of effect here in the double bond of the molecule, and in the remaining 2 phenols, but in much less manner. And lastly, we have the radical attack. As you can see, the attack is very focused on this phenol, on the same phenol, but the region is dispersed all through the second aromatic ring, which it was proposed that it would aid in electronic dispersions once the covalent bond was formed. And well, the 2 remaining phenols also have these effects. So what can we conclude from this? We can conclude that resveratrol has an antioxidant effect, which grants fidelity to our results and validates our methodology. Then that the BCMPA esterification increases hardness in various steel veins, and it was confirmed with other previous work from Dr. Bermo's research group. That the unprotected bees show positive effect on solubility such that it can be used as an exclusionary factor when assessing molecules reactivity. And finally, that B2 bees, due to its potentiated reductive capacity and electron accepting power, once associated with hardness, we could see that the electronic donation would occur, but only to the limits of the valence electron. So he would grant it an anti-radical activity. Thank you.
Thank you, Miren. Does anybody in the audience has any question? You can raise your hand. Yes.
I have an ignorant question, so hopefully you can help me out. So you do mention that it is a very computationally intensive, right? Very, very much time consuming and so on. But you're working with DFT, correct? And my understanding was that compared to other techniques, for example, such as Hartree-Fock and so on, it was actually less consuming and more efficient. So I was wondering if you could, shed a bit more light on that part.
Well, yes, we have previously worked with Hartree-Fock, and it is indeed less accurate than DFT analysis, but it does less take time. It's much rapid, the process of Hartree-Fock than that one of DFT analysis. This took like, I don't know, like 8 hours, 2 hours. Well, it depends on the sample, but from like 24 hours to compute. So it's possible to have much more time, but it's hard. Yeah.
But I just want to hear a little bit.
Yeah, but DFT is a much more precise method, to do this.
Thank you. On Zoom, we have a question, please, Dr. Juan.
No, sorry, I just keep my hand raised, but it's wrong. Thank you.
Okay. There's a time.
So those were some beautiful figures, Miren. Congratulations on everything you've done. And I have a question from the other side or the other perspective, but you mentioned that you used water like a solvent because you are thinking about biological applications of these. So could you elaborate a little on how you plan to take these molecules that you found, for example, the anti-radical activity for, and bring them to the biological applications you want to use them for?
Well, okay, so we don't have any super decided path because it's still on the theoretical step of the project. But since it has some proven anti-radical behavior, we would like to experiment on different cells and known radicals, especially reactive oxygen species that produce in the cells that have many damaging effects. So we see from tumors to damage to oxidative damage of the cells. So we would want to see if these molecules could counteract that effect. And actually, the thing that was verified with BCMPA has been approved in some treatments and in some molecules to be used in humans. So that would be a first step to know that we are heading in the right direction. That's why we choose that one specifically. Yes.
Thank you. Dr. Kathy.
A small suggestion, because I got lost at certain points, put the structure, please. I saw, no, not now. When you when you put the table, put the structure, because I saw million of times these structures, but I cannot correlate them between the structures and the name. And I have one thing, because you know me, I'm tickismicky when it comes to organic chemistry. You made example of the LUMO and HOMO, and you say that you're using a dienophile, and this resventol is the LUMO as a dienophile. Pay attention, because to be a LUMO, you need to use electron donating groups. Electron withdrawing groups, while the OH is an electron donating group. So you're speaking about an inverted disaldered reaction where you're not using the dienophile as the LUMO, but it's going to be the HOMO. So just to pay attention, if you're presenting it in the future, that is just.
Thank you, Doc. Any other question in the audience? No, in Zoom? Okay. So seems that we are done. Thank you so much, Miguel. Okay. Do you have a plan to bring your hypothesis into an experimental model?
Yes, we are planning on taking it the next step into an experimental model. We will start working on it in the course of the next year or so, maybe. Just as I mentioned, Dr. Sataray, our main project is for human applications, so we are going to get there eventually. Thank you.
So please give another applause to Miren. Okay, audience, now we will have a 15-minute break. So we will see you back then at 4:15. Thank you.
We are coming back. Next is choose person. It's a join. Let's start, but yeah, let's go back.
Okay, now we are going to see two presentations of one project divided. The first one is DFT approach to the reactivity of curcuminoid analogs. The undergraduate senior student Josué Guzmán Cabrera presents a study concentrated on the theoretical evaluation and analysis of two different curcuminoids derived from the organic framework of curcumin. His mentor was Dr. Fernando Belmont. And the other one is discovering the power of curcuminoids, synthesis and characterization of enhanced antioxidant capacity. The undergraduate senior student Nadua Alejandra Pineda Nájera presents a study in which she explores the reactivity of two curcuminoids not yet reported in the specialized literature, labeled as bromocurcumin BC and aminocurcumin AC. Her mentor was Dr. Fernando Belmont.
Thank you, everyone. I hope everyone can hear me fine in the back. If not, let me know. My name is Josué Guzmán. I will be talking to you about DFT approach to the reactivity of curcuminoid analogs. So a little bit introduction. So what is DFT? As you probably become experts, because I've seen a lot of presentations with this, but the DFT stands for density functional theory. And this is a theory that's used currently in quantum chemistry as it reduces the computational power by a lot, as it's able to solve the many body problem. What is that? We see here, we have 3 electrons, 1 ion. We all agree we have X, Y, Z axis in a 3D plane. And the calculations will have to be made for each X, Y, Z axis. So this one, with this system, would have about 12 different calculations. Now imagine systems that are a lot bigger in terms of electrons, nucleus. It's not gonna be viable to use standard methods. So what DFT does is it models electrons as a cloud, hence the density part, density function, and it won a Nobel Prize in 1998. So curcuminoids in DFT. So curcumin, this is the structure, comes from a turmeric plant, Curcuma longa. Maybe if you're from like the Southeast Asian part, you eat it constantly in the food. But the important thing to highlight is that it's been studied for a long time, and it's proven to relieve oxidative and inflammatory diseases, such as those on the board. And most importantly, for our purpose, is the structure, which is divided into 3 parts. We have 2 rings, one here, one there, and they're connected by a bridge, or what we call a bridge. And we can see these functional groups. We have an O with a stick and an OH. So what we want to do is we want to change that a little bit and have a different structure in terms of functional groups. So that's what we set out to do. So this is what a curcuminoid is. We've been able to synthesize 2 of them, bromo and amino curcumin. And we wanted to use this for photodynamic therapy, as it has been proven to be useful in photodynamic therapy and in terms of treating certain kinds of cancer cells. And recently, Dr. Belmont published an article about this. So we're using that as the foundation for our research, or sort of like a continuation, a bridge, or a future direction. So what are we proposing? We're proposing to investigate the electronic properties of both the aminokurk and the bromokurk, take into account that the bromo is expected to be an electron withdrawing group. while the amino is expected to be more of a electron donated group and it's sp3 atom. And so these are the reactivity indexes, which you've probably been familiarized already, but if not, we're using chemical potential, chemical hardness, and chemical softness, which are related as they're the inverse of each other, but you can follow the equations in case you have any questions. We have electrophilic index, which is an index that measures how electrophilic our compound is to be, meaning how many electrons it can receive. And the maximum number for electron is correlated to that because it's going to give us an insight into how many electrons our compound can take if it is electrophilic. And then lastly, we have the nucleophilicity index, which is the opposite of electrophilicity index. It shows if you have electron density and you're able to attack or donate electrons. So our objectives again is to calculate the reactivity indexes that we've seen earlier using the frontier orbital tool theory, which is why we saw energy in the calculation or in the equations. We saw the energy of the HOMO and the energy of the LUMO. Highest unoccupied molecular orbital is the HOMO, and it is going to hold the electrons that are most likely going to react in a chemical reaction. While in the LUMO, we have the lowest unoccupied molecular orbital, and that will have an energy that's the energy required for an electron to jump into that molecular orbital. So what we did. So we can sort of like break it into two spots. First thing was we used Gaussian. Gaussian is a software used similar to Orca, which we've seen in previous presentations, that does all these kind of calculations in terms of quantum chemistry. We were able to optimize the structures of aminocurcumin and bromocurcumin using the B3 lip function and the basis set def2 SVP. The importance of the def2 SVP is that the P part stands for polarized, meaning that it takes into account the polarizability of our compound, and so that's why we decided to go with that basis set. And as well as this was sort of laid out in Dr. Belmont's previous publication. So we kind of just went off of that one as well. And then for the second part, for the HOMO and LUMO energy calculations, we decided to use Minnesota 15, which is a functional that does the TDDFT, time-dependent density functional theory. And with that, we were able to calculate the values of the HOMO and the LUMO energies. So here are results. So this is for the bromo structure. On the right side, we have a simulated UV spectrum, which is simulated because we got these from the calculations that we were able to obtain. And on the left side, we have an electrostatic potential map, which just highlights the distribution of electron density across our molecule. If you see a blue color, it means that there's low energy or low electrical density there. If you see green color, there is electron density there. And if you see red, that means there's a concentration of electron density there. And the importance of this one is it can be used to be characterized. Our group, our project, we use a different software for that, but the simulated UV absorption can be used to characterize our molecules as if you go into the lab and run the UV spectrum test. We should be able to see a peak around this wavelength, but you'll see why it's important to have this as well later on. And then here we can see that we have a lot of blue throughout the whole molecule. I mean, sure, we have some green in the bridge on the O's, which should be expected because it's an oxygen, so it's electronegative, but there's still a lot of blue, suggesting that this could be more like of an electrophile. If we compare it to the amino compound, we can see that our molecule, our bridge, lights up, flares up beautifully with a green color, red color, and this suggests that this would be a better nucleophile instead of a electrophile. And similarly, we have our simulated spectrum, UV spectrum. So then here are the actual hard raw values. The ones that are highlighted, which are on the bottom, the lower HL gap, a less negative mu, lower energy or lower value of hardness. These are all the characteristics that we want for a photosynthesizer for our photodynamic product. And those can be, those are the ones that are highlighted or in bold. That's the group that took that category, perhaps. And we can see that across the board, the aminocurcumin basically took it all. So it suggests that it would be a better candidate, better suited for to be a photosynthesizer for photodynamic therapy. We can see that the electrophilic index. We have a higher electrophilic index for bromide, which makes sense if we take a look at the electrostatic potential map, which I mentioned there was a lot of blue. I mean, sure, there's green, but the color that predominated was blue, suggesting it was more of, it was lacking electron density. We have chemical hardness. We can see that curcumin, the amino curcumin takes this one, and that if it's soft or if it's harder, means that it's harder for the electron density in the molecule to be manipulated. So what are our conclusions and our future directions with this project? So as I've mentioned already many times, the bromocurcumin displayed a higher chemical hardness, suggesting that this would be a good anti-radical instead of a good antioxidant, which is what we would require for a photosensitizer to work with photodynamic therapy. And that the aminocurc exhibits a higher absorption. And this is relevant because in photodynamic therapy, what you do is you can apply the topically like a cream, or you can inject it intravenously, and which is the part of the cancer. Then we shine a light, an LED or a laser at a certain wavelength, which activates our curcumin. This would cause our electrons to go from a singlet state to a triplet state. And when they come back down, they would release energy. That energy would affect the oxygen found in our bodies, creating reactive oxygen species that would interact with these cancerous cells. And so a higher wavelength is correlated to less energy. So if we need less energy to excite these electrons, it's a better candidate for a photosensitizer. To further continue this research, we want to continue to calculate these changes in the functional groups, but now try different halogens to see if we have a periodic trend, from fluoride being the most electronegative in the periodic table downwards to iodide, and see what kind of effects this has on the electrophilicity index, the chemical hardness or softness that this can provide, and how this can be put in a table or characterized and see which one is better photosensitizer. And lastly, there is a clear tendency that we can observe with the aminocurcumin being more of a nucleophile, because we saw the electrostatic potential map, which was light up green and red compared to the bromocurcumin. And here are references, and I want to thank Dr. Belmont, Nadwa, because we've been working this for a long time, and everyone else that helped me along the way. I also want to thank Arkansas State University for allowing us to use their computers and their facility to conduct this research. Thank you.
Thank you, Josue. Now we will proceed with the questions for this specific part of the project. Does anyone in the audience has a question for this part? In Zoom? Okay, so no questions?
I actually have a question, and you allowed me to ask you this because you asked me just a maximum of bromine. So you say before, and then actually on the bromine, you say before bromine is electron withdrawing or electron donating?
We expect it to be electron, even though, speaking, it should be an electron.
Exactly. So are you planning, for example, to make a comparison with one that is actually an electron donating, like paramethoxy group, so anisole groups, starting from the anisole and synthesizing and trying that one, so you are sure that is an electron donating group, and making a comparison, because bromine is actually electron donating. No, yes, electron donating, and no, electron withdrawing, sorry. But you are expecting in this case to act as an electron donating, isn't it?
We expect it to act as an electron withdrawing.
And
so
it is electron donating, and it's what you are expecting to act.
correct. Yeah, and that would be a good idea for further down, but we also want to try the halogens to have those affected. But you'll see with Nadua's presentation, the actual in-lab results so that you can correlate that even though it should be an electron donating group, it actually acts the opposite. Yeah.
I want to not leave it out. Okay, so now we will proceed with Nadua's presentation. Please, Nadua.
No. ¿Me puedes tomar tiempo?
Okay, so good afternoon. My name is Nadua Pineda, and I'm going to present the second part of the project that is discovering the power of curcuminoids, synthesis and characterization for enhanced antioxidant capacity. So first of all, as my colleague Josué told us, curcumin comes from the curcuma longa, but when we extract this part, we obtain a mixture of curcuminoids. We have curcumin, demitoxycurcumin, and bis-demitoxycurcumin. In this case, we are only going to focus on curcumin as we have 77% yield of this compound. So it's the one that we are aiming for. In this case, what we want to do is to change, as my colleague told us, the analogs to see the reactivity it can have. Curcumin, it has a lot of anti-inflammatory capacities, and it has been able to use it for the treatment of certain cancers. The problem is the viability this compound has. As in the body, the consumption, the absorption of your body, it will only take 25% of the curcumin for use it in your body. So it's something that we have to take into consideration, and we have to modify this structure to be able to change the viability and help us to improve it. So we have the synthesis. In this case, we decided to do two salialdehydes commercially available. This one is the dibromo, and this one is the diethylamino. These two are available in outreach, and the synthesis that we performed, it was from the salialdehyde that we can see here. The X and the Y will be aimed for the bromos, and the A will be for the diethylamino, and the B will be for the hydrogen. So making an aldol condensation, we have a base, and we added the acetylacetone, and then with some acid, we were able to obtain the curcuminoid template. So in this case also will be bromo, and in this case will be the diethylamino, and in here the hydrogen. And for this one, the bromocurcumin, we obtain a yield of 66%, and for the amino one, we obtain a yield of 55%. So it's a really good yield comparing to the first ones we obtained. Then we have the characterization part. In this, we did the UV-Vis spectroscopy for bromo and amino. We're comparing it with some calculations that we performed, but in this case, we did it with this basis set, but with Orca software. In this, we can see that we have only a 0.032% absorption difference. So this means that we are really sure we have the compound. We did the theoretical part or the calculations to have like background about the structural part. So in this case, the blue one will be the theoretical, and the red one will be the experimental that we obtain. So this one is for the bromo, but we have to tell you that we have an imaginary frequency of 4.14. That's something that we have to work in the future to get rid of it. Then for the amino, we have a 4% difference in the absorption. So it's a little bit higher, but it still is in a good range. And in this one, we have two imaginary frequencies. So also it's something that we have to work on it, but we can see that it's a really good tendency that we have for the structure of the molecules. Then the melting points we have for aminocurcumin from 40 to 41 degrees Celsius, and for bromocurcumins from 79 to 83% degrees Celsius. Then thanks to Dr. Basurto and Bachelor of Science Alison Suits, they help us to make this ferric reducing antioxidant power FRAP technique. That this one is to measure the antioxidant capacity they can have, or the antioxidant power some compounds they can have. In this case, we have for the aminocurcumin 0.045 average of micromole Rolex equivalents. And the problem that we obtained with bromocurcumine is that using the solvent 75% ethanol, we were not able to obtain it or to perform the experiment. So the future work that also we have to do in this project is to find a solvent that they can help to measure this for the bromocurcumine. But the importance of this is that we have antioxidant power in the aminocurcumine. That is something that we were aimed for. And then we have the cyclic voltammetry. In this one, this experiment, what they do is you have your compound, your solution, and you will be able to oxidize or reduce it, depending on the capacities or the characteristics the compound has. So in this case, we have the bromocurcumine, and as you can see, we have 3 cycles. You can see a little lump in here, and in here, and in here. This lump indicates that this compound is reducing. So it's something that our colleague, Josue, was telling us about the theoretical analysis, that as a bromo, it's hard and has electrophilic capacity, then it's going to reduce, and it's something that we can see in here. We have, this one was able to obtain thanks to the Dr. Rafael Rios. And we make a solution of bromocurcumine of 10 milligrams over milliliter with an electrolyte of calcium nitrate with a 0.5 molar solution in a 101 mixture. So this is something important in bromo. And for the amino, we can see the opposite, that also it's something that Josue told us, that the amino will have the nucleophilic index, so it will be able to oxidize. So in here, what is happening is that we have this like little lump in one here, and we have one here and one here. So it means that it's oxidizing, but it's not only oxidizing once, so it's oxidizing twice. In this part, also we use the same concentration for the curcumine, and we use the same solution for the electrolyte in the 101 mixture. So this one, what they tell us is about being a confirmation about the activity indexes that Josue was telling us. So for the conclusion, what we have is that there are some commercially available aldehydes that we can use to synthesize new curcuminoids. For the aldol condensation was the better one that obtained us a high purity of curcuminoids. We also have that there's an evidence for a correlation between the energy predicted by DFT that Josue told us with the experimental values that we obtain. And for future work, what we want to do is to synthesize a portfolio of new curcuminoids and also check for the reactivity they will have. So that's all. Thank you for your attention. I want to thank Arkansas State University for providing the tools and the equipment for being able to do this research. And I want to thank Dr. Belmont and Josue for make us part of the team. And that's all. Thank you.
Thank you, Nelva. Now we will proceed with the questions. Please, if someone has a question regarding this part or the whole project, raise your hand. Or maybe in Zoom, do we have any question? Okay.
No, I have one.
Okay.
So I've seen the new synthesis that you tried it, but just to say for the ones that maybe doesn't know, this is implied that there been several students working in that synthesis. That synthesis needed to be optimized and so on. So can you explain us how many different kind of synthesis did you try? Because I remember that there been boron involved, there been some other reactions there involved.
Yes, so we only had like 10 minutes to present, so I obviously took all of the work that didn't help us. But in the synthesis, first we had a problem because we were not able to get the curcuminoids in the solid phase. So they were only in the aqueous phase because we think or we predict it was because it was like attached to the boric acid. So what we were doing was performing a lot of different techniques or experiments or changing some values to check if we were able to get it for the aqueous phase to the solid phase. But we were like doing it for several months and with several or different kinds. Like we decided to use the rota vapor first, but that wasn't helping at all. So then we decided to let it ride like naturally or do it other things. But that's how we were able to obtain like this synthesis or this, yeah, to perform it. So that's a lot of work. Yeah. Science. But yes, yeah.
Thank you, Nada. Any other question? Okay, so since we are done. For questions, one for you, one for you. Thank you. Now we will continue with a presentation under the title Nest Site Characteristics, Diet and Activity Patterns of Cirrus oculatus in a Mixed Forest. Undergraduate senior students Pablo Torres, Jimena Zamora Prieto, and Diego Emilio Aznar Cruz examine nest site characteristics, diet, and activity patterns of Cirrus oculatus in an oak pine forest in Querétaro. Their mentor was Dr. Nicolás Ramos Lara.
Okay, so hello everyone. It's okay. Hello everyone, I'm Jimena Zamora, and I'm presenting this team project called Nest Site Characteristics, Diet, and Activity Patterns of Cirrus oculatus in a Mixed Forest. And these amazing people here, and this is my team. So a little bit of context. The scientific name of this arboreal squirrel is Cirrus oculatus. Peter squirrel is the common name. Louder? Okay. The common name is Peter squirrel in honor to the person who discovered it. As I said, this is a tree or arboreal gray squirrel, and proudly it is an endemic species. It can be found in pine oak forests or mixed forests. And this type of ecosystem can be found all along the Trans-Mexican Neovolcanic Belt. So basically, this is the distribution of the species. Why we conducted this study in the first place? Because there's a severe lack of information about the ecology of this species, especially about its behavior. Also, squirrels are forest indicators, so the more abundant squirrels are, the better forest quality. And because there's a discrepancy in their conservation status, because as MRNAT says, this is a protected species, and IUCN says this is a species of least concern. The objectives are, of course, contributing to the information about the species, particularly about the nesting behavior, activity patterns, and dry season diet, and all of this for preparing better conservation plans. This study was conducted in El Bosque de las Hojas, that is a private pine oak forest located in Cadereyta, Querétaro. It was conducted in 2022 from April to July, that is a dry season, because the highest temperatures recorded in Querétaro are during this period of time. The methodology is divided in three. the information about nests, information about the trees where the nests are, and squirrels. So the information about nests included height, materials, condition, position, orientation, and support about each nest. What we needed to know about each tree included coordinates, height, using this Pavlov clinometer, species, condition, basal diameter obtained with the DBH measuring tape, connectance and canopy cover, that is basically tree density, obtained with the densitometer and a compass. As this was an observational study, we did not interact with any squirrel nor with any nests at all. So all the observations were made with the help of binoculars and with this ethogram over here that we designed, including all the information that we needed to know about each squirrel that we see. Now, the results about the nest site that include nests and trees. And after quantifying all the observations that we obtained, we found that there is no tree preference for nesting between pine trees and oaks. These two are the most common trees for nesting. And in this chart here, we can see that pine trees have a little higher percentage, but this value is not considering some factors. So we applied the statistical analysis, p-value, to obtain the significance. And as this is not significant, this means that pine trees and oaks are equally preferred by squirrels for nesting. The reason for this is because pine trees provide more food but less nest materials, and oaks provide better nest materials but less food. Then here we have average and standard deviation of these parameters that we needed to obtain to establish a general pattern. And according to the p-value here that shows significance, we found that squirrels prefer to nest at the top section of the canopy. The reason for this is because the higher the nest is, the better. Because ground predation is more risky than aerial predation. So the higher the nest is, the safer for the squirrels. About activity patterns, we found that this species shows morning behavior and supported with the p-value that shows significance. And here we have this circular graph showing all the hours of the day. Here is the mean. And in this section over here, we can see that during morning hours, squirrels go out from their nests. And they prefer to go out in this time of the day because environmental temperature is lower, so the hydration risk is also lower. And most sightings were in branches, and the most common behavior is arboreal traverse. This means that, and we can see the graph here, graph of behavior, and that this squirrel spend the most time running and moving all around the trees. Something very exciting that happened during this study was that we recorded the first mating chase of the species. I could talk about this the whole day, but you can see the full video here. It was recorded in May when we spotted one female and some males with scrotal testes, and we followed them. And they led us to an area with many squirrels that were running and playing, jumping, chasing each other. And it was a beautiful event. and that's how we got mating behavior information about these species. And when it comes to the diet, in April, we found traces of pine cones or pine seeds and strobiluria, that is pollen. In May, we found a lot of apples, of beaten apples by squirrels, of course. And in June, we found other seeds and mushrooms in addition to the other food. And by July, we found the first signs of acorns, indicating that wet season or rainy season was about to start. And also we got new information about this squirrel because they like to save their food in between tree branches. Okay, just to conclude, we said that this squirrel shows no preference between pine and oak for nesting. They prefer high and thick trees, nesting at the top section of the canopy in highly connected trees. They save food in tree branches, and during dry season, they eat mostly pine seeds. They are mostly active during mornings, and arboreal traverse is the most common behavior, and we have mating behavior information about the species. Here are my references, and thank you so much for your attention.
Thank you, Jimena. Does anyone in the audience has any questions for her? Yes, doctors.
Go ahead, you go first.
So, Jimena, first of all, and I've said this multiple times, congratulations to the team. You did an outstanding work, and I know that you did a lot of camping for all of this. I'm going to make an ignorant question again, because I was looking at your standard deviation values. I don't know if it would be possible to maybe show them again. And as I said, it is an ignorant question. I want to know if these values, or if you will, the percentage error in a sense, is what's common in your field. Let me give you an example. In astrophysics, it's very common to have errors of up to 40%, and it's rather common. I would like to have an idea of whether the errors that you get or the standard deviations that you get, if they're common in your field or not, because I don't know.
Thank you for your question. Actually, yeah, it is common because there's a lot of factors to consider when you're working in the field. For example, maybe we missed a nest, or maybe we missed a squirrel. We were looking at one squirrel in here, but maybe we are not looking at other that was in another place. So yeah, that's why we prefer to use a p-value just to obtain the significance. But yeah, the margin of error, yeah, it's always there.
Yeah.
Okay, another question coming from ignorance, because I'm not a biologist. In terms of the diet, how do you distinguish that the seeds and the things that you showed, they were actually eaten by the squirrel and not from other animals there? So what is how do you make that distinction?
Thanks, doctor. Actually, the teeth of the squirrels leave a mark, a specific mark, like two marks in the seed or in the apple. The apples are the most obvious ones because you can see like two lines doing this. Yeah, it's here. Here, these are pictures that we took in the field. So, and here you can see like two marks
and here another two, and there were many like that. So yeah, that's like an indicative that it was a squirrel.
Thank you. I think we have another question also. Okay, yes, of course.
This can go first. So I will say it again, congratulations for this fantastic work. I think it's very complete. And I think it's fantastic that you went through the effort to do the statistics, because that's tough. Now, I precisely wanted to ask about that. So you have that table where you have your results for the preference analysis that you did. Right. So there you say there's no tree preference between the two species. What is that based on? Like, what would be your null hypothesis, or what would it look like? Oh, yeah.
Thank you. Okay, every time we see a nest, we also consider the tree. And actually we have a graph of many tree species. And the most common ones were pine trees and oaks. So in between these two, and according to the data that we obtain, like based on the nests, we got that value.
What I'm getting to is the proportion of versus so that's what you would use as to build that model. Right?
Yeah.
Because it seems that it's very close to 50-50.
Yes. Yeah, and again, maybe there was one nest in one oak that we didn't count. So probably that's why this percentage is a little lower for oaks.
Yeah. So on the same subject of preference, you also have that diet information later on. And in theory, you could look at preference, right? Based on that data. Have you guys thought about doing that?
No, we didn't consider that. We just we first we had to obtain like what's their diet, and yeah, then a second step would be like making the statistical analysis of preference for diet. But yeah, we didn't get that far.
Yeah, what you need to measure is like the availability in that region of each of those items.
Yeah.
Whether they like.
And with the amount of evidence about the diet that we found, maybe we could have done it, but we didn't.
What's that question online? Oh, there are many questions online. No, two.
Okay. This study was realized in a private forest. Yes, it was a private forest called El Bosque de las Hojas in Cadereyta, here in Querétaro. And
I think it's more than one. So
okay. How much time?
It's okay. Yes.
Behavioral data will never have a standard or normal error that we should expect, especially because we are working with different organisms. Rather, the data distribution will say whether out of it is. Yeah. Thanks for that comment. It is true.
Here another one.
How can these results be extrapolated to a forest open to the public, which is surely most exposed to human interference? This study was conducted in a private forest, but it is open to the public actually. So many people can go there and put their tents on and make a camp there. but the forest, like the trees and the area, the vegetation is not affected by humans. And this is one of the main rules of going to this place.
Okay, we have two questions on Zoom. Please, Mr. Gerardo Arias first.
Hello, can you hear me?
Yes, we can hear you.
Okay, very nice presentation, very interesting. I've never seen before a work or research that has been done with squirrels. So yeah, this was very interesting for me. I was sort of looking to your results, to your statistical results and your p-values. Could you tell us which statistical tests did you use for your different variables?
Yes, well, this statistical analysis is called p-value, and basically it's the probability that based on the observations that you made, like the probability of assuming that the null hypothesis is. And in the article, we included chi-squared, and it is not here in the presentation, but yeah, in the article it is too. That is for non-parametric data.
Okay, that is then the statistical test that you used.
Yeah, also we use the Rayleigh here, VR. This is when you're working with angles.
Because the p-value is not, in fact, it is not a test, but it is actually the result of the statistical test that you are using. So in this case, for instance, if you used a t-test, I don't know how many observations that you have, for instance, about how many times did you see the different trees, for instance? How many observations on each tree? Do you remember?
Yes, it was 17 nests in each tree. And yeah, we used chi-squared because the data that we got is non-parametric. I believe that t-test is for different data.
Okay, thank you very much.
Thank you.
Thank you, Jimena. Just another question from Dr. Juan, please.
Hi. Statistic in the nature is really difficult, so we can get less errors if we can use some things that are used around the people that live there. You can use trained dogs, and you can look for more nests and squirrels by that. So maybe you can try it. Thank you.
Thank you. We will think about it. Thank you.
Hello, this is Pablo Torres, is one of the other team members of the project. And just expanding on the answer to Dr. Miguelena's question about some of our statistics, as Jimena mentioned, we did use chi-squared for most of our statistical analysis, not only because it's non-parametric, but also because it's continuous data. And our null hypothesis that we were testing against in this case would be that the distribution of the nests would be equal among pine and oak trees. So it would be a 50-50 distribution, which we found to be the case. In our results, it was nearing 50-50 distribution, which means that there is no preference for the squirrels for the different genus of trees. Availability of trees would come into it, but we didn't consider this data for the analysis. And we're out of time.
Thank you. Thank you very much, Jimena, for.
Our next presentation is titled Animal-Human Conflict, Cheetah Conservation Efforts in Africa and Iran. Undergraduate junior students Fernanda Cabrera Moreno, Sergio Eduardo Vaca Castellanos, and Magda Figueroa Diaz present a review in which they highlight the significance and consequences of the loss of this species. Their mentor was Dr. Nicolás Ramos.
Thank you for that lovely introduction. So, like she said, we're talking about the animal-human conflict of the cheetah and the conservation efforts done in Africa and Iran.
So, due to the recent cheetah population decline in countries in Africa and in Asia, the conservation of these animals has become a crucial thing in the ecological communities. So, according to the IUCN, there's been a 30% decrease in the total population in recent decades. Also, conservation efforts in these countries that we were going to be talking about have been mostly unsuccessful due to various aspects, mostly being the loss of habitat and human-wildlife conflict.
So the objectives of this review are to highlight the conservation efforts done in the African countries and Iran, to address the success and failure of such conservation efforts, to know which one is the most effective in preserving the genetic information of the cheetah, and lastly, to know how these efforts can intervene with the agricultural practices.
For the methods of this review, we had sleepless nights with caffeine trying to find these research papers. And in order to address the problem, which is the conservation efforts, when we're talking about conservation, is usually very gray. There's no one way or the other. So we wanted to highlight the major problems since this population is so small. The history of the cheetahs spans back all the way to 4 million years ago in North America for some reason. There was a North American cheetah that supposedly migrated from North America all the way to Asia, all the way to Africa. I said supposedly because they thought that it was a cougar until a few years ago, and they redefined that it was actually a cheetah with genetic research. All of the species went down in the last ice age, 10,000 years ago. All of the species population plummeted, causing the extinction of the North American cheetah, as well as a 97% population loss in everywhere else, which, as you can see, left us with a lot of fragments in Africa and in Iran. The yellow is the historical range that they had, and the darker yellow is where they are actually now. So because of the decline in their population, we had something called a bottleneck effect in the genetic variation of these animals. And that is one of the conservation problems that we have today. Today, the population is still declining due to human interactions. Shocker. Poaching, habitat loss, all of these things come into account. And the problems with conservation is that we need to try and save the genetic information of these animals before they go extinct.
So the biggest country that is involved in the conservation of these animals would be Botswana, which has at the time the biggest number of individuals, with an estimate of about 2,500 individuals in the whole country. So what are the challenges that these cheetahs are facing in the country? Well, the main ones, like I said, would be habitat loss and fragmentation, animal-human conflict in the form of poaching, and conflict with the farmers who protect their livestock from these animals. And like my partner said, would be genetic isolation due to the absence of corridors between the populations in the country. Also, what you're seeing here is the logo of Cheetah Conservation Botswana, which is the organization that is in charge of protecting these animals in the country. Their efforts they do is mainly a program that has had a 100% successful succession rate, which is the Livestock Guardian Dog Program, which involves training guard dogs to protect the livestock from these animals, as well as community outreach and education programs, the establishment of protected areas, and the laws and regulations established within these areas to protect these animals. But despite the improvements that the conservation of the cheetahs has had in the country,
there's still a lot of work to do to maintain this animal's long-term existence.
So there are two similar papers that talk about the conservation efforts in Namibia, but they're addressed differently. One talks more about the density of the population and the other one more about the behavior of the cheetah. So overall, the main threat is the human conflict, as in Botswana. Humans kill cheetahs to protect the livestock from predation. But I found one paper that says that they analyzed the scat of the cheetahs and they found out that the livestock is not really part of the diet of the cheetah. So humans just kill cheetahs because they think they're mostly a threat. So a challenge is that even though there are 140 reserves, these are not suitable habitats for the cheetah, and they're fine surviving on the commercial farmlands. Just to point out, the density estimation is of less than 2 cheetah per square kilometer. And regarding the conservation efforts, the Cheetah Conservation Fund did the same thing as in Botswana with the dog program, since it was successful. And ecotourism has also been found really successful, having visitors coming to the farmlands and getting to know about the cheetahs and wildlife. In Algeria, the situation of the cheetahs is terrible. There was a study done in the Yahagar Cultural Park, and that is of the size of more than 600,000 kilometers square. And they placed 40 camera traps for a span of 23 days, and they recorded the animals that were passing by. So this graph, you found the 8 species that were found in the camera footage with its relative frequency. Just 1 cheetah was found twice in the camera footage. So this is bad because Algeria has the greatest concentration in the North Africa. So this means that the population is either declining or the home ranges are changing.
So in Iran, we have the most isolated population of cheetahs that there is, counting with less than 50 individuals in the Dasht-e-Kabir area or province, which is the one circulated in the image. The Asian cheetah has lost over 80% of its historical range in the country, so there's not a lot of habitat left for these animals. And due to these tiny populations, the greatest threats that they face would be genetic ones, which will be genetic isolation and inbreeding. Also, the hunting of their prey, the overhunting done by the population of Iran, affects these animals in the sense that they need a lot of energy to sustain themselves. They are mostly muscle, so they have no fat stores to retrieve energy from when the food sources are low. So the overhunting leaves them with a reduced number of prey to consume and overall reduces their fitness. So like I mentioned, most of the methods of conservation that are being done were in the Dasht-e-Kabir area, which contains these 2 national parks. The one would be the Turan Biosphere Review, which is the one in red, and the Kabir National Park, which is the one in green. In these parks, they have the deployment of rangers that protect the individuals themselves. They also have compensation for livestock in the adjacent areas, which are mostly farmlands, as well as breeding programs to prevent inbreeding and the restore of habitat, well, their historical habitat, sorry. The most successful of all of these would be the breeding program, which includes breeding these animals with cheetahs and specimens from all over the world, especially from Europe and from reserves in Africa.
In all these factors in the conservation, we have one common factor, which is the fragmentation of the whole thing. If you can tell, in the southern part of Africa, there's a large concentration there. However, that span crosses 6-7 countries, and the communications between these countries are not great due to their conflicts inside of the countries. So there's a lot of problems here because the cheetah has a very large home range, one dating from 3,000 kilometers to 1,000 kilometers. So they need a lot of land in order to do their hunting, their breeding, all of it. And since it spans many countries and a lot of cattle and all of these things, humans see them as a threat because they see big cat with big teeth. So they rather kill the cats rather than actually just leaving them alone. That's why the education needs to be done, and that's why we need to focus more on poaching and habitat loss. Next. In this paper, we see the protected versus unprotected areas and how with protection, obviously, they don't go extinct. Who would have known? And on the other one, we see that there's different countries with different kinds of conservation areas. Obviously, we have higher conservation areas in Africa in some areas. However, at the bottom, we have 318 kilometers. That's nothing for the cheetah. We need at least 1,000, if not more. So we need to up those numbers a little bit. Next.
So the conclusion would be that the greatest threats for these animals are habitat loss and human interactions, whether it be poaching or conflict with the farmers. And the main issue in creating a unified plan for these animals is that their home range crosses a lot of provinces and a lot of countries who have disagreements with each other and political problems outside of conservation. Also, all of these conservation efforts have had mixed results and efforts are still very necessary. So the results have said that the conservation is possible, as well as the coexistence with us and with the animals that also inhabit these areas. It's still rudimentary, but it is possible. Thank you.
Oh, do you guys have any questions? Do you guys have any other questions about the review? No? I thought she was gonna say it, but I guess I'm doing it.
So any questions for the team? Yes. We have a question here.
Hello. Great presentation, thank you. My question is just about the conservation company or organization that deals with trained dogs, with training dogs. I just want to know how that works, how that functions, because I found it very interesting.
So basically what they do is that they first get in a certain breed of dog. They didn't disclose the breed of dog. They just said it was hunting dogs and sheep dogs. It could be a hybrid of both of them. They're not pedigree dogs or anything. And what they do is that they basically have them in a little program or campaign with volunteers that train them to not hurt or fight with cheetahs, but to just circle around cattle. It would be like just normal cattle dogs that keep an eye on livestock. It would be the same as they do for wolves, but here they are trained to identify the scent and the looks of a cheetah, and that is the danger that they need to protect the cows from. And they don't actually, like I said, attack the cheetah. They will just take the cattle somewhere else. Have you seen how the guard dogs guide something like that? They would just take the cattle somewhere else within the range, and the barks and the noises will alert the farmers that something's going on, so they can also take a step in. So that's how that works. It's completely done by volunteers with the organization. I hope that answers your question.
Any more questions? I think we had one in
I
Zoom.
have a question in Zoom. Can you explain the methodology for your review?
The methodology of the review, I don't understand. How we conducted the review, is that the question? How we conducted, we wanted to focus on conservation and we thought, big cats, let's see which ones are in trouble. We went to the IUCN and we saw that the cheetah was critically endangered, right? Critically endangered. So we wanted to focus more on that because, like I said, 7,100 individuals are in the wild. So that is a very low population size for any kind of species. So it is very important to keep their genetic diversity up. And okay, nice. Anyway, but yeah, we wanted to go to focus on endangered species, and we just fell on the cheetah because they're pretty. Any more questions? Dr.
Yes, I continue with the question in the chat. Hello, I'm Dr. Pavel Lugo. And my question is around about the organization of your information. You made a correct explanation. This is a really good compilation of your data, but you don't analyze this information. You need to analyze. And my question is around about how you organize this information and why you don't analyze this data to obtain yourself conclusion about this investigation. Am I explained?
Yes, it's because we only had 10 minutes. We only had 10 minutes and we could have put all the graphs and all the organizations there. For each country, there's one organization and they're especially in that one country. And then they talk to the other countries and then there's an overall one with other set of numbers. We used we have the numbers in the references and we could have talked about them. However, we only had 10 minutes and we just wanted to highlight the problems in the conservation efforts.
Okay, I see more important the analysis of the information of a known problem than the compilation of this data. Am I explained?
No, I didn't totally get that. Can you repeat the question?
Yes, for you and for the audience is more important the analysis of the information more than the compilation of the talk about the existing problem that already we know that exists.
Okay, yeah, we'll take that into account for the next state that we get going to. You had a question?
Anyways, thank you for your presentation. Very interesting. So in particular, you were talking about the Iranian population and the breeding efforts that they're doing. And I think you mentioned that there actually includes like breeding them with individuals that are kept in, in other countries and in European zoos and things like that. So is there a risk there of losing the genetic diversity that is particular to that population when you are breeding them with other individuals?
Well, yes, there is the possibility of losing the the particular genetic diversity of the Iranian population. But since the numbers are so low, the priorities right now with the organizations is to just keep the population going. The Asian cheetah is another way to just mention is the same species of cheetah. The location is just different. So the only particular thing about these animals is that their location in per se. So right now, what is important to the organizations is to keep the species alive rather than just the diversities between each of the species of the different places. I don't know if I'm answering your question, Doctor. Okay. Thank you.
Good time for more questions. Okay, we are done. Thank you. Give them a big applause, please.
Oh, okay, thank you. I love plants.
Now we will continue with a presentation under the title Correlation between captivity and glucocorticoid levels increase in penguin species. The undergraduate junior student Alejandra Rodriguez Barquero presents a review for understanding how captivity affects the behavior and physiology of certain penguin species and the actions humans can do. Her mentor was Dr. Nicolás Ramos.
Thank you. So yes, I am Alejandra and I will be presenting the correlation between captivity and glucocorticoid level increase in different penguin species. Let's begin with what are these lovely creatures. Penguins are flightless birds. They only live in the southern hemisphere. They mostly live in the Antarctica, but they can also be found in New Zealand, in South Africa, in the Galapagos Island, or in Peru, for example. They possess flippers instead of wings, which make them flightless birds. They have dorsally black plumage and ventrally white. This counter shadow provides the camouflage for their predators. Also, they vary in sizes. Some of them are smaller, some of them are larger. What is the objective of my review? Personally, because I really love these species, I think that it really is important to understand the negative effects the captivity have on the behavior and the physiology of all of these species, not only in penguins, but in all the animals. Also, the action humans can do at zoos or at aquariums to ameliorate the captivity situation. So why penguins? Why did I choose penguins and not other animal? Besides that I really like them, I think that it's really important for them because they are really important in different ecosystem roles. They are the predators of smaller species and they are the prey of larger ones. They fertilize the landscape. This is known as the ornithogenic impact. This is because the feces of penguins have high levels of carbon, of phosphorus, of nitrogen, so they fertilize the landscape. Also, because of their wide habitat range, they conform a lot of species in that area. It has been observed that humboldt species at the top and Magellanic, for example, they are really easy to train species, so they can be easily found in different aquariums or at zoos, for example. And finally, they make us smile, well, at least me. So moving forward, what are glucocorticoids? Maybe you have heard of them. Maybe you say, oh, maybe I know what they are, but not have a clear explanation. Glucocorticoids are steroid hormones produced by the adrenal cortex, which are necessary for life, health, and disease. They are the responsible of the regulation of different homeostatic functions, such as metabolism, cell division, or even development. The thing is that these levels increase by circadian rhythms. It is normal, but stress is a factor that it is really affecting this increment. We have two types of stress hormones, cortisol and corticosterone. What's the main difference? It is really simple. It's basically the same. Cortisol is found in mammals and corticosterone is found in birds. So that's the main difference. It can be the increase of these levels can be natural or induced. What do I mean by natural? For example, if a penguin is hungry, if it is cold, if it's been predated, these levels, of course, will increase. The induced ones are these stress that they are not accounting with. For example, captivity. All of these, natural or induced, have negative effects. Our final term to define is captivity, which is really important. Captivity is defined by the Cambridge University as a situation in which a person or an animal is kept somewhere and it is not allowed to live. It is really important to highlight the point that it is not allowed to live. We know that there are captive breeding programs where the main goal is to, maintain a species, don't lead to extinction. What's the problem? That in some few of these places, the animals are treated in the best optimal conditions that they can be. Sadly, in most of them, they are treated not as good as they should. The problem, the lack of mental and physical stimulation. This is, of course, because of the reduced space the animals are developing. They can be cages, they can be fish tanks, for example. Reintroduction failure. The problem is that because these animals they do not know how to live in the nature, when they are reintroduced to the natural habitat, it is more likely that they will not survive. Finally, morphological effects. This is really dangerous because, for example, in tigers or in lions, because they have been provided them like with ground meat, like already chewed, these animals lack the morphology in the jaws. What do I mean? They lose the strength and they lose strength in their paws. If they are reintroduced to their natural habitat, it's mostly likely for them to starve. And so what did I do? Basically, after I knew what my topic was, I jumped to the DNBLS library provided by the school and also through Google Scholar. I looked for my keywords, which in my case were captivity, glucocorticoid, and penguins. Nevertheless, some of them were kind of useless, so I focused on the important ones. My range was from the 2010 papers to 2022 papers, looking for their reliability. Finally, I distinguished the key ideas, the papers that work for my investigation and one does not, and the one that does not, and organized the information for me to present. So after a lot of investigation, what did I found? In chronic captivity, the chronic means a long-term captivity. The effects are really bad. The mass body condition and morphologies is being really affected. Either the animal lose a lot of weight or gain a lot of weight. Also, a glucocorticoid level fluctuation. This means like a variety of these levels, and most of the time they stay really high. The problem, if these levels do not go down to the normal state, the immune system and the reproductive system is being a lot compromised. What happens? The animal is more susceptible to have different diseases or even to never reproduce. Also, adrenomedullary effects, which are really bad, negative. The worst thing is that they are permanent. Therefore, they can lead to the death of the organism. So moving forward with more penguins, how the presence of humans affect different species of penguin. I will just say the normal name because it's easier. So Adelaide penguins and yellow-eye penguins, they have reduced breeding success. Adding, the yellow-eye penguins have fledgling weights. This means that the animal is losing their feathers. So the thermal conditions is being compromised. Humboldt penguins, which are the ones that are mostly found in the aquariums, they show an increased heart rate, which led to an increased blood pressure, which is really dangerous for us humans and imagine in penguins. Also, the king, the gentle, and the royal penguins have shown a different behavior. They actually become aggressive. Increased vigilance and agonistic behavior, they start to fighting, which is really bad. And finally, in the one that I found the most concerning one is the white little penguin. They avoid nesting areas. What does this mean? They are not even planning to reproduce, which can lead to the extinction of this little species. This has not only been observed in penguins, it has also been observed in different animals. For example, I bring the Eurasian red squirrel. As you can see, in the x-axis, we can see the hours post-capture, looking for 24 and 32. And in the y-axis, fecal GC metabolite. We can see a relation when the animal is just captured after 24 hours, the glucocorticoids in both female and male tend to increase. If these levels do not go down, we have serious problems. Also, not only in squirrels and in penguins, we can also observe this in jaguars, for example. Okay, so don't get loose. The NEX means the No Extinction Organization, and the BC means Brazilian Zoo. The black bars means high visit density, and gray bars means low visit density. So for example, here we can see a clear distinction between low and high salivary cortisol. So the presence of humans is also affecting the jaguars in general. What is the take-home message of this review? Stress is inevitable. The animal can get stressed by normal things, but it is our job to make the animals to suffer the least as possible. Also, people start to care about. Years ago, the owners of the aquariums just care about selling tickets and do money. Now they start to care about the well-being of the animals. So for this, it is really important to give the animals the best conditions for them to live happy and to survive. So if you want to check any of the things, here are my references, and I will be really happy to answer some of your questions. Thank you.
Thank you, Ale. Does anyone in the audience? Yes, I'm coming.
I'm gonna let the professor ask the question.
Oh, it was a great presentation, first of all. So again, out of complete ignorance, but as a person that has been to aquariums many times, no, one of the things that was a bit of a red flag from what you mentioned is that I've been to aquariums that have specifically Humboldt penguins, and they show the people that they have had success in breeding the animals, right? That they reproduce, and they show it some sort of evidence that the animals are well taken care of and they are completely comfortable in their breeding, right? But now you mentioned that actually that's not one of the side effects seen in Humboldt penguins. So would it be safe to say that this is not a good indicator that the animals are actually in good conditions in these places? Yeah, the breeding.
Okay, so, well, if the animal is like breeding, it means that they are comfortable. For example, I don't know the aquarium that you went, but, okay, I was going to talk about that one because I'm from Veracruz and one of my uncles, he's like well, not the president, but he has like a high range in that thing. And I asked him when I was doing this presentation, I asked him like, the increased heart rate and all that stuff. And he mentioned that actually the penguins in that aquarium, they have like a resting space, like at the back where people cannot see. So when a penguin is getting like so stressed because of the whole people that visit that aquarium in a day, they can just go back, like chill a little. And also they have their nest there. I don't know if you remember that you can see like these holes in where they are. So they are their nest. So every time that they get like really stressed, they just go to their nest and just like relax themselves. Also, these penguins are like taking them and like, well, they don't take them outside, but they are like training them. Like training these species make the stress levels to like go down. So yeah, the Veracruz Aquarium has this program, like breeding success is a total accomplishment because they now have like this problem of having a lot of baby penguins that they don't fit. So this is a good indicator that the penguins are like really nice, really good. So if they are not like interbreeding, that would be a red flag. That would be a really problem. So yeah, so that aquarium, for what I know, for what I've seen, because I have wondered, they are doing like nice things. Yeah. 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 Well, at least for that speech. Okay. I read? Okay. Just as a suggestion to Alejandra, whenever you are using graphs and figures from other authors, the authors of the figure should always be acknowledged in the slide. Oh, okay. Yeah, I take it for the next presentation. Thank you. Someone else?
Okay. Any other question among the audience? Yes, Doctor.
When I went to the aquarium in Veracruz, actually, in order to receive the penguins, that's the explanation that I have remembered is that they should have meet certain conditions in order to receive these ones. The question is if you are aware of other programs, for instance, for different species to be implemented. So I don't know if you are aware of certain conditions or certain programs for other species that are not the Humboldt ones that are in place, for instance, because I think that Veracruz Aquarium is really successful because what I remember is that the Humboldts, they were donated by another aquarium somewhere else, but in order to receive the animals, the aquarium should show that they have the conditions to have the animals in good care, right? So I don't know if you are aware, according to your investigation, if there are similar programs in some other place regarding other species. So that would be my question.
Okay, so what I have heard that my uncles told me is that, for example, in the Humboldt or even in the penguins, in the penguins or even in the dolphins that they have there, their food that they consume, for example, the penguins, they bring the sardines all the way from Peru, that they don't buy it in Superama in Veracruz, no. They went all the way from where they used to live. So they're giving them like these very special conditions also for the dolphins. although these dolphins were like captured in Veracruz seas, they go to that specific place and like fish those specific fishes. Honestly, for other organizations, I am not aware of, but I can totally investigate and I can send you an email about that, and that will be my pleasure.
Yeah, I mean, because it would be interesting. What the
Mhm. Mhm.
and check.
Yeah. Oh, you're welcome. Thank you.
Explain the innovative factor in the review.
Basically, what I did find like the innovation is that people, as I said, they're like starting to care about now, not only on these like fashion species, they are start to care about their physiology and how the animal like can live in the best conditions, so they can whether live without any stress and also have like these breeding programs. So yeah, the innovation, what I think is that one. People are starting to care about, and people are starting to notice these changes if you do like the right things about not only penguins, as I said, all the animals that maybe can be found in aquariums or even in zoos.
Thank you, Ali. So that's it. Thank you so much. Sí, pero lo pones aquí, ¿no? Pero primero más. Oye, tú no vas a cambiar. ¿Tú la vas a cambiar?
The next topic presented will be gene therapy as the key to cure sickle cell disease. The undergraduate junior student, Emily Sabrina Colunga Correa, presents a review on gene therapy and its different methods, focusing on genetic sickle cell disease. Her mentor was Dr. Nicolás Ramos Lara.
Well, hi, my name is Emily Colunga, and as I say, I'm going to present gene therapy as the key to cure sickle cell disease. I would like to start this presentation by asking you a question. Actually, 3 questions. The only thing you need to do is raise your hand if it's a yes, and keep your arm as it is if it's a no. Let's start. Do you know what is gene therapy? Okay. Do you know what is sickle cell disease and how it works? Okay, great. Do you know how these 2 things work together to cure? Okay, no worrying. That's what I was expecting. So let's start. Basically, my objectives of this review is to inform you about this type of topic, how this will cure sickle cell disease and how it's already been done, but it needs improvements. My basic objective is to inform overall because I think informing is saving time and is creating interested. So don't worry, the ones who say no to any of the questions, I'm going to answer it for you. And the ones who say yes, I'm going to rephrase the information. So please, next slide. What is gene therapy? In simple words, gene therapy is any type of, is when you put corrected information to a gene that presents symptoms of a disease. Gene therapy can be done in 3 types of way. We have lentiviral, adenoviral, and retroviral type. Here we're going to focus on lentiviral. Basically, we have here a lentiviral type of cell when the most important thing will be the RNA because it's the one they're going to help to encode to a new thing. So let's start. Here we can see in a stem cell how the lentiviral factor will enter the stem cell, will pass to RNA, to then transcription to DNA, and then will enter the hard part of the nucleus and then do a transcription. Now, what is sickle cell disease? Sickle cell disease is a multi-system disease and is specifically in the B globin gene and is more specifically in the 17 nucleotide of this gene because adenine is chained to thymine, meaning that they will make crystallization. Here we can see in this image, a normal red blood cell, we have a circle, and a sickle cell, we have a moon shape. This will cause crystallization in veins, meaning that after some white oxygenation would not be able to be, and this crystallization would cause damage to a lot of organs in our system. How do they work together? They have worked together with a simple technique of, instead of doing the normal type of therapy for this treatment, that is grabbing the stem cells and put it in a blood transfusion and do it, they change this. They grab the bone marrow of the patient, they edit the gene to a stem cell that is healthy, and before the third step, they do chemotherapy to wash every impurity they can have in their system. And after this, they condition the bone marrow of the patient again to be able to accept the new stem cell. And the fourth phase is simple. They just insert the treated stem cell into the patient and hope the modification is done correctly. This technique have already worked, but it has worked with some, bad results and some good results. The biggest clinical trial was 17 patients. We can see in this graph, the yellow, orange little line is the patients before treatment. And the little line in the middle, we can see we have blue. The blue are healthy patients in general. And we have the green one, and it's the patients after the treatment. They show better and less deformity in their red blood cells. In this case was 1 year complete of treatment and 12 months of follow-up. They have a problem in this case. They have 2 cases of leukemia after the overall process, but this was explained with other article that said that the leukemia wasn't because of the gene therapy. It was because in part of the therapy, some off-target DNA was reached and activated a gene that it was predetermined in the patient, that it was leukemia. We have benefits and risks of this type of gene therapy, of course. Some of the benefits is that it is less invasive. It's best to have just 2 years of your life with needles or having all your life with needles and drugs, because you can't have a good quality of life with that type of, disease. This also will decrease a lot of health risk, just such as getting problems in your glands or not being able to have a normal life overall. And it will lower your need of immunosuppressive drugs, because immunosuppressive drugs affect all your organs and also will affect lower your defenses. So if we take off this, you have less possibility to, die in general. And the risk of this type of therapy, most of the risk is the chemotherapy. The chemotherapy will, of course, weaken your immune system, meaning that you are more, you will be more up to having different diseases that can activate when you are in your lower defenses. Also, we don't have money to do the research. So the problem with this type of therapy is that a lot of factors around this type of therapy haven't been researched because of money. We also have the fact that this can get you cancer that has been proven right but wrong. Because if we had more money, we can correct this because we can stop the off-target DNA to be activated. It's really high cost. That's the real problem because right now, that type of therapy is $1.85 million for just one patient. So you can afford it. It's not a normally affordable treatment, and it's not approved by the FDA. The FDA has approved trials for this clinic, but hasn't approved it because we only have, example, in the USA, only one clinic that are studying this. So we cannot approve something that haven't been studied enough. The importance and my goal of this review is to inform you and make this topic more interesting to people, because a lot of people don't know about sickle cell disease. A lot of people don't know about gene therapy or are afraid of gene therapy because they think, oh my God, if I get gene therapy, I will grow another foot. But of course that won't happen. That will be weird, that it's a misinformation. So I would like to inform to you about this type of topic. And because informing is saving life, and informing, getting more interesting, can also give more horizon. For example, you in the back, let's think that your mother is a scientist. You go to your mother and say, wow, I learned this topic today. And then you might go to your boss, hey, I learned this topic today. And the interest and information will spread, meaning that it will be easier to people to like doing this type of research. And with the problem of the money, if we don't have enough horizon, we're losing new techniques. The newest technique is theoretical, and is, can you pass to the next? Using CRISP to stop the chemotherapy needing. It's called a tree of tactics, and it's basically grabbing a mutated B globin there is going to be a study in vitro, and then applying it and put it above the stem cells, and then just inserting it in the gene. In that way, the big globin will like eliminate the other big globins that are mutated and just modify and adapt in the blood. And also sending the reinforcements is highly important. They trying to do like micro healthy red blood cells to then put it in the treatment and be able to send it and be easier for them to recover and well adapt again and to mix with the unhealthy blood. Also bypassing big globin, that it will be instead of grabbing just a needle and put it, grab the crisp scissors, cut it and put it directly to the big globin that is already been modified. And as good as new is like the backup plan for this theoretical meaning. They will be basically do a really mini gene sequence in vitro and then applying to the patient directly. The problem with this is because we don't have a lot of money. Because we don't have a lot of money, it's not possible yet to investigate CRISP, neither CRISP, neither gene therapy. So that's the biggest problem, fundraising. Thank you so much for your attention. Here are my references in case you want to scan them, and thank you for your time. Do you have any questions?
Thank you for your wonderful presentation. Do we have any questions from the audience?
How much is considered the cost of one of these therapy? Not about the research, about the therapy.
The therapy is evaluated approximately in 1.85 million and is one of the cheapest therapies for gene therapy in general. So it's not affordable and it's only done in clinical trials because it's not that affordable. Another question?
Yes. What is the prevalence?
We don't have a direct statistic for Mexico because it's a really unknown disease here and a lot of people don't know how to differentiate the symptoms, but it has been said that in Latin America we have 2% of the population who can have it, like 1 in 1 million will have it. Another question?
Oh, that's a question. Raise up.
Okay, can you ask your question, please? iPhone.
So is the raising hands, the person that is using an iPhone, if you want to open your microphone and ask the question, please now.
Thank you. This is Dr. Medina. A nice research there. Do you think that you are injecting the gene therapy just in blood, or they are looking to put it in the bone marrow so we can change the cells where they are growing?
They are actually putting in the bone marrow and the semi-treatment is in the blood transfusion. So yeah, they're already like thinking about doing that, but because the problem with the bone marrow is that you need to condition the patient to be able to accept it. So that will be a lengthy process, but they're trying to do it, but it's a little risky. So they are just starting with the first trials of that type of therapy.
Yes, like transplant, bone marrow transplant. So
yeah,
yeah. Okay, thank you.
Thanks to you. Other question? Thank you.
Thank you again, Emily. Our next topic is the reintroduction of the harpy eagle in the Mexican territory. The undergraduate senior student, Victor Hugo Larrieta Hernandez, presents a proposal for the reintroduction of this bird to Mexico with the intention of avoiding their extinction in nature. His mentor was Dr. Nicolás Ramos Lara.
Testing. Okay, good afternoon to everybody. I want to ask one simple question. Do you know anything about this eagle? No? Good. Harpia. Let's see, where can I begin? Where can I begin? Okay, he's a bird of prey of almost a meter tall. The female ranges from 90 to 95 centimeters tall. The male has sexual dimorphism between half of the height of the female. The wingspan reaches almost the 2 meters. They are the biggest eagles in all America. Oh yeah, the claws are almost as big as a grizzly bear's. The talons are as strong, very strong enough to break a human bone, especially the radius and the ulna or a leg. They are especially prepared birds of prey for monkeys, sloths, children of 5 to 4 years. No, don't worry, they don't attack humans. Next, please. Okay, the objective is more than nothing. It's more of a personal thing for me because I love these birds. I had the luck to see them and handle one in this arm. It was quite awesome for a kid of 6, 7 years old. But being honest, the reason for doing it more than personally is because they have a cultural benefit and they're part of Mexico's history. They're almost in some murals in Minatitlan, Veracruz, in Oaxaca, Tabasco, Chiapas. And these are apex predators. So they are controlling the presence of monkeys and sloths, which are eating the leaves or eating the fruits out of the trees. So by having them present, we keep the erosion or the loss of the trees to a minimum. Next, please. Distribution. As I always said, they are mostly present in South America nowadays, Nicaragua, Costa Rica, Brazil, Venezuela. But in the past, I'm talking like in 1960s, 1950s in Mexico, they were present in all Veracruz, Oaxaca, Tabasco, and Chiapas. Yes, now in the wild, they can only be seen in Minatitlan only. And that's because they have a specialized zone for them to nest, which has its own festivity called La Festividad de las Aves Migratorias, the festivity of migratory birds. That is in Minatitlan, October 5. And in Chiapas, I think it's October 8. All this information I was obtained from San Diego Zoos, which are the leaders in keeping them alive in captivity, in breeding. They are they're quite, well, this is the map nowadays. As I said, Brazil, Venezuela, Colombia. Okay, in Mexico, it is possible to observe the need, as I said, in Chiapas, especially, what is Concordio? Here, I think it's Concordio, Chiapas, Ocosingo, that is closest here. And Ocosotla, Espinos, Chiapas, that I don't know where it is, sorry. I'm in Minatitlan, here in the center of Veracruz, close to Tabasco. Okay, the dangers and threats. Obviously, humans. Why humans? These eagles don't fear, they don't have a fear for humans. They just like, oh, I see a human, let's go see him. Because there's no other predators that can kill them. Just look at. Human activity, habitat loss because the deforestation and the non-protected use of the resources. There are fewer prey due to the loss of their habitats, climate changes, and unfitting nest sites. They have a specific need for area and a specific tree, which I think I remember correctly was a chestnut. and subtropical habitats also. Look how cute. Now, solutions. I thought first in a solution by myself, which was by using the help of BID, the Banco Internacional, Interamericano de Desarrollo, in relocating them in Quintana Roo, thanks to the searches of Dr. Monroy Ojeda in where's the harpy eagles. He said that there was a possibility of relocating them in Quintana Roo. Why? Because they keep also the presence of the trees they need, the area. But the thing is, obviously, politics in Mexico are not in favor so much for us, the biologists. A way to contrast the fact of the politics and not wishing to use the money, the other thing that I was thinking about was using the help of the World Bank to support the idea, seeing if it's possible to be helped and being in movement by the help of them. These are my references if you wish to see them by yourself. Questions? Yes.
I have yet another ignorant question. Exactly, because we're all scientists, we don't feel uncomfortable saying that, right? You mentioned that you propose to relocate in Quintana Roo because it has, the appropriate characteristics for this eagle to thrive. But because it's a predator, would it have a negative impact on other species that are in Quintana Roo already?
Basically, basically, the harpy eagle is a hunter specialized for mammals and certain types of birds that are mid-sized called macaws. Almost they will never reach out for a small sized. I try to do the specific check. There's usually in Quintana Roo, there's no that much birds that can be eaten or hunted by the harpy eagle, exactly, because they are faster than them. The harpy eagles are canopy hunters. They are not really fast enough to catch the toucans or other birds. They are more specialized to hunt the monkeys, sloths, especially the three-digit sloths, and so on. So there's not that much possibility of risking a lot of Quintana Roo because most of Quintana Roo is now being used for hotels. Someone else?
Are there any other questions on Zoom? And Dr. Pavel says, is it appropriate the introduction of an organism in another habitat different of their natural ecosystem?
Well, usually it's not appropriate to reintroduce other species in places that they shouldn't be. But Dr. Monroy, in his research, was speaking about the possibility of reintroducing them because there's not that much land or woods to support let's say 200 or 500 species members of the species, because especially in Minatitlan, it's been on the risk of entering more humans into the spaces that are designed for this bird. Chiapas, it's also the case. They are moving more people, and it's almost greater and greater impossible to stop from polluting the river, especially in Caño del Sumidero, which is where they're usually flying. because there's a presence of monkeys and sloths there, especially. I don't remember crocodiles, but I don't think no. But returning with Dr. Roy, who was speaking about there are a presence of possibilities of introducing them because they cover everything that is needed for the eagles. And it shows there is not a huge impact, just it will help to keep them safe because they are wild extinct. If we can say they are extinct in Mexico almost, and only the inbreeding help of San Diego zoos has been keeping them not extinct. They are being protected. Do you know how many individuals there are in the wild? How many in captivity? I don't remember with certainty how many they are in the wild or how many are in captivity. It was said in the page of San Diego's, but I don't remember correctly. So sorry, I don't have the answer. Anyone else?
Thank you so much, Victor. Here you go. Are the students Dante, Didier Lopez, and Maria Fernanda Estrada present on Zoom? No. I think that was all for today. The students were presenting strategic management, the digital tool for learning and strategic concepts, and mentored by Dr. Saul Esparza and Dr. Steve Lacey. Thank you very much.
We are closing the oral presentation section. Tomorrow we're gonna have the poster presentation. Unfortunately, we don't have the opportunity to have a Zoom streaming of this event. So whoever is gonna be in campus, we're gonna enjoy for sure this event. Thank you very much.